Hi class, in this second video on infectious diseases, I'm going to talk about the production of monoclonal antibodies and what they're used for, vaccination, and HIV. So this video addresses assessment statements 11.1.1, 5, 6, and 7, and assessment statements 6.3.7 and 6.3.8. Okay, so in this first assessment statement, it says, describe the production of monoclonal antibodies and their use in diagnosis and treatment. So a pathogen will have many antigens on its surface. So if you want to have an antibody, have a set of antibodies that's specific for only one antigen, not all of them, you inject that specific antigen into a mouse, and then that mouse produces cells that have only that one specific kind of antibody in it to that specific antigen. And the problem is that these cells will not stay alive outside the body for a very long time outside the mouse. So you fuse them with a tumor cell, and tumor cells stay alive a long time outside of the body. So, and when you fuse them together, you make what's called a hybridoma cell. And this hybridoma cell lives a long time outside the body, but it also, you have an endless supply of monoclonal antibodies. So you lyse them and then you can harvest them from these hybridoma cells. Now, what would you want to use monoclonal antibodies for? Well, your book discusses two of them. One use of them that your book discusses is sensing the human chorionic gonadotropin protein, which is a hormone, and it's only produced during pregnancy. So if you think you're pregnant, you would do one of those tests with that you pee on the indicator and it changes colors. And usually what happens is that they have the antibody to HCG attached to some kind of color changing enzyme, and when the protein is present, you see a change in color, or you see a plus show up, and that would indicate that you would be pregnant. Alternatively, you can use it in cancer cell recognition. So there are different antigens that are presented by different cancer cells, and if you can determine which antigens are present in a certain kind of cancer, you can take a blood sample and test for whether that antigen is present in your blood, indicating whether you have that type of cancer or not. So essentially, monoclonal antibodies allow you to be very specific in the kind of protein, kind of foreign protein that you're sensing for, that you're trying to detect. In the next assessment statement, it says, explain the principle of vaccination. So you have an organism that has these proteins that are being presented on the surface, and they're called antigens. These are the things that antibodies can bind to. What you do is you break up this organism, either it could be a virus or it could be a bacteria, and when you break it up, it's no longer alive, but you still have these antigens present, and you produce antibodies to these antigens, and by injecting this into some other host. Usually for vaccinations, it's usually eggs, and then you harvest the antibodies that are produced by the white blood cells. And now these antibodies can bind to the antigens that are presented by the pathogen. In the next assessment statement, it says, discuss the benefits and dangers of vaccination. So firstly, you should see here on this image on the right that the benefits of vaccination vastly outweigh any dangers that are presented. So you can see in each one of these cases, as we scroll down here, we can see that the number of cases in the United States decreased dramatically as a result of vaccination. So if we take a look at smallpox, right? The number of people that died before right? 29,000 odd. And the number of people that have died since the vaccination era for smallpox, none. No one has died in the United States after we introduced vaccination. And if you look for tetanus, about 500 people died in the pre-vaccine era, where the number has decreased to 14 in the post-vaccination era for tetanus. So as your book discusses on page 289, you can eliminate the disease. Elimination of diseases as a benefit. You can decrease the spread of epidemics. So these diseases that can kill a huge number of people can be decreased or totally eliminated. You are doing preventative medicine rather than medicine of responding to diseases. And preventative medicine is cheaper than trying to deal with someone who's already sick. And essentially, when you're vaccinating somebody, you're giving them immunity without experiencing the symptoms, since you're giving them either a dead version of the pathogen or a version that has been significantly weakened. So we should talk about the dangers as well of vaccination, which are pretty minimal. Prior to 1999, vaccines contained thimerosal, and thimerosal is a mercury-based preservative. Thimerosal is mercury-based, and mercury is a neurotoxin. But after that, no vaccinations contain thimerosal, so that is no longer a concern. There's a perception that when you put multiple vaccines into a child, you can overload their immune system. But that has really been dealt with because the antibodies that are produced as a result of vaccine are much more specific than they used to be. That the number of antigens that are presented to your immune system is much smaller than it used to be in vaccines. There's been some anecdotal evidence of an autism link, but that autism link has been shown to be non-existent. And then there's allergic responses in a very small subset of people. And that can usually be predicted as a result of an allergy to eggs or the production. So that is pretty small. So assessment statement 637 says, outline the effects of HIV on the immune system. So HIV and AIDS have a wide variety of effects, because basically the HIV is compromising the immune system. HIV prevents the immune system from creating a full-fledged immune response against other infections. So people don't die of HIV and AIDS, they die as a result of other infections that can't be battled because of HIV infection. So the next assessment statement says, discuss the cause, transmission, and social implications of AIDS. So AIDS is caused by HIV, and you can see in this image, first the HIV virus binds to a T helper cell, the host cell, which is a T helper cell. It fuses to it. The RNA, which is inside, gets introduced into the T helper cell. It takes the RNA and makes its own DNA, and that's using an enzyme called reverse transcriptase, as you can see here. It's taking the RNA and making the DNA in the reverse direction from the central dogma. It takes that DNA, and the DNA gets integrated into your chromosomes. And then it uses the machinery in your T helper cells to create RNA, to make protein, and then to create more viruses that are released from your T helper cells. And if you remember from the previous video, T helper cells are involved in cloning cells that have recognized antigen. So they're important for antibody production. And if you are not promoting the mitotic divisions of cells that recognize antigens, you cannot fight the pathogens that enter your body. So once you infect your T helper cells, they can't clone and they can't stimulate antibody production. So then you can't fight infections. The next part of this assessment statement says to address transmission. So, firstly, you can transmit HIV through unprotected sexual intercourse. It can be transmitted from mother to child during delivery and through breast milk. And you can also transmit HIV if you share needles, and this happens with intravenous drug users. And very rarely it can happen through blood products. But usually the blood systems, especially in the United States, are screened so that this is extremely rare to get HIV in this method. In the last part of that assessment statement, it says, discuss the social implications of AIDS. And this is a very large topic which people have wrote books and made movies about. But briefly, because HIV and AIDS was originally to be found to be prevalent in homosexual and drug user communities, there was a stigma placed against people who had it, even if they had gotten HIV and AIDS through other methods like blood transfusion. There was a serious stigma against them, and of course, funding these issues was initially very difficult. And of course, the prevalence of HIV in these communities really led to people being not treated properly, not being treated medically, and especially in places where there's inadequate medical care, like in sub-Saharan Africa, where there's not as much education about how the disease is spread, how the virus is spread. There can be larger outbreaks of HIV and AIDS, which kind of compounds on itself. And even though the disease rates in the United States have decreased, the disease rates in these areas in the world, but also in poorer areas of the U.S., has still either been maintained or increased. And so it's a problem for all of us to address. And we'll be discussing some of that in class. So if you have any questions, please ask. Otherwise, I will talk to you soon. Bye.